Pritha, can you please start? Your video is not on.
So now?
No. Yes. Perfect. But the screen is not visible. Yes.
Okay. So, very good evening, everyone. I am dietitian Pritha Biswas on behalf of Cognize Nutrition. Today, welcomes you all on this insightful session of role of nutrigenomics in handling chronic diseases. I welcome everyone, our speakers, our host, our moderator, as well as our entire dietitians fraternity. Now, I would want to introduce the today's host, Shomindu Ghosh, who has 17 years
You have
of
to change your slide, Pritha. Yes.
who has 17 years of experience in the field of clinical nutrition and dietetics. He is also a renowned diabetes educator and lifestyle counselor. Currently, he is leading various online courses for dietetics and nutrition students and professionals. He is practicing as freelance dietitian and attached to Soulful Steps. He is also attached with NGO SOS Village to provide nutrition education to rural mothers and their children. He was formerly associated as dietitian lead in Eastern Region, India for Abboud Nutrition. He also worked as a diabetes educator and clinical dietitian at various hospitals like AMRI, Park Clinic, KPC Medical College and Hospital. He has also participated in pan-India operational research to study outcome of diabetes in terms of HbA1c and quality of life in diabetic patients conducted by IDE in New Delhi at Apollo Multispeciality Hospital. He has also worked as a corporate lifestyle counselor at various organizations like IBM, TCS, Vodafone, Reliance, Jet Airways, etc. He was also involved in nursing training program for different hospitals and also worked as a nutrition counselor for more than 500 school children with various organizations. He also worked as a coordinator and speaker in diabetes education in various eminent organizations as well. He is a live member of IDA, IAPN, and the live member of Association of Diabetes Educator. Now I would like to hand over the entire session to our host and respected sir, Shomindu Ghosh. Over to you, sir. Thank you.
Thank you, Pritha. And it is really privileged to have Dr. A. Srikumar and Dr. Banerjee in this platform of the Dietitians Fraternity. And this is all possible because of Mr. Rupak Banerjee, who is long-term, we are a friend, or he is like my elder brother. He's a very dynamic person. So I came to know about him when he was in BD, and he was the very well trainer in Forum for Injection Technique, that is FIT. She was very enthusiastic. She was a coach. She has worked for 25 years as a regional manager, and he has varied experience in diagnostic, nutraceutical, diabetic, metabolic, oncology, radiology, wound management, etc. So he has traveled in many countries also, and he was looking after India, entire India, and Bangladesh. So I welcome Mr. Rupak Banerjee to introduce our speakers for the today and give the introduction, because we are eagerly waiting for this enriching topic. It is nutrition and nutrigenomics, which is the upcoming topic for the nutritionist, because medicine, as you all know, the people are more dependent on the medications. People have erratic lifestyles. We need to change their lifestyles, and depending on the genes, we have to treat our patients. So this is a very modern outlook for all of us, and we are very much eagerly waiting for this session and more physical session in Kolkata. So I invite Mr. Rupak Banerjee, and I can change the slide, please.
Thank you. Thank you so much, Shomendu. And it's really a very nice way of organizing in a very short span of time. I thank all the participants who have already joined. I can see more than 92 participants in the portal as of now. So it gives
I think we are not able to hear Mr. Banerjee, right? So
Yes, yes.
because of the network glitch, so just give us some time. He will again continue. We have many senior dietitians and nutritionists from our faculty, so it will be really enriching. Yes, Rupakda, you are back. Yes, you can continue. You need to unmute. You need to unmute. Yes,
Yeah,
please.
I have unmuted. Right. Can you hear me now? Hello?
Yes,
Yes, yes,
yes,
yes,
you are
yes.
audible.
Right. So once again, good evening. Sorry for the little bit of a technical glitch. We are very privileged to have two very important scientists with us. So I would first take the opportunity of introducing Dr. A. Sri Kumar, who is the person who coined the terminology Nutrigenomics in India. And he brought in the new concept of precision medicine with the help of integrated Shomendu, sir is unable to hear. Dr. Sri Kumar is unable to hear us. Have we muted Dr. Sri Kumar? Shomendu?
Hello. Yes.
Dr. Sri, you can't hear us? Hello? Dr. Sri is probably not getting the audio.
Sir have to again join in. Sir have to
I will just one
Yeah,
second.
just text him.
I'm
Rupak, text him.
Please request sir to rejoin.
So I once again welcome you all, and I take this opportunity till the time Dr. Sri Kumar rejoins the forum. Dr. Sri Kumar is the founder of BioWell Hospital, as well as he is the founder of Integrated Functional Medicine Hospital in Kochi. And he is a trained head and neck surgeon with an ENT background. And still, he has a functional experience of more than 22 years of functional medicine. Sir, can you hear us now, Dr. Sri? Hello?
Yes, sir. Your voice is listening.
Just one second.
You need to unmute.
Unmute, sir.
Rupakda, I think some mode has been changed for Dr. Srikumar's mobile. We can see if it is possible, if he can join from his mobile also, then you can share the screen, or we can share the screen if it is possible.
I am trying that. I will do that. Just one second.
Otherwise, Dr. Banerjee can continue some discussions with us. Sir is back, but the same mode is showing. Rita, we can see a symbol in the right side of doctor's picture, so that thing, I think some mode has been changed in his device.
Yes, sir. Actually, my screen is share, so I cannot see anything from laptop. So I'm seeing from mobile itself.
Or one thing that can be done, doctor can speak from his mobile and screen can be shared from his laptop. That can be done. So he have to join from both the devices. Off করে দাও যে।
Sir, I think sir have to close the companion mode from his phone.
Companion mode, where is that companion mode?
Sir, as you the host, can you do that for sir?
Companion. If I can change, acha, just I am making you host. If you can do the favor. I have made you host, Pritha.
Yes, sir.
Sorry everyone for the technical
Somendu,
problem.
what I suggest till the time Pritha goes ahead with this, let me introduce Dr. Banerjee.
Okay.
And
Okay.
we can start with Dr. Banerjee's presentation for the time being.
Okay. Okay.
Pritha,
So-
could you go to the next slide?
Yes.
Yes, sir. Sure.
So it gives me immense pleasure to introduce Dr. Banerjee. Dr. Biren Banerjee, who is a scientist, but more than a scientist, I would say that he is a teacher, he is a leader, he is a great motivator and orator, and over and above, he is a human geneticist. So it is better that he speaks and we all get enriched by what he speaks on the program today. Let me introduce and welcome Dr. Biren Banerjee to spare some words of his wisdom with us. Dr. Banerjee, please.
Thank you, Rupak. Can I share my screen?
Yes, sir. I'm
Sir, please.
closing it.
Yeah.
Yes, sir. Now you can share.
Can you see the screen?
Yes, Yes, sir. sir.
Is my screen visible to all?
Yes,
Yeah,
sir.
yes,
Perfectly
sir.
visible. You can.
Okay, so good evening, everyone, and thank you for this opportunity. First of all, let me thank Rupak for organizing this meeting, and Somendu, the entire dietitian community of Calcutta. It's great seeing about 100 plus participants on a Saturday evening. So I'll be talking very few things. I'm not a dietitian. I'm basically a student of genes and DNA. So I'll be talking to you about how DNA and diet can be related and how we can be gene wise. That's the topic I have selected for this evening. I belong to the city of Bhubaneswar, and which is now India's #1 smart city. We have defeated 98 cities and became the top contender of smart city. So I'm proud to be a smart city citizen here. And we have slowly, in the last couple of decades, we have made a transition, or still making a transition, from cell-based understanding of our physiology to DNA-based. So that the diagnostics is moving from cells to DNA. And thanks to the Human Genome Project, cracking the code of DNA of 3.4 billion bases of DNA has been one of the greatest achievements of humankind in this century. So when the Human Genome was published, Bill Clinton told that now we know how God has written the book of life. But actually, do we know exactly how God has written the book of life? No, because the answer is not no, not yes, because the book of life, the chapters written in 23 chromosomes are dynamic sentences. They keep on modifying by our environment. So there is epigenetics in the play. So if DNA is the software, who writes the code? We all must ponder upon this fact that the DNA is the software, the blueprint of every life, from bacteria to man, from elephants to grasses to plants to sea. All the biosphere of this world is having a software encoded in the DNA. So that is the programming language of life, but this programming languages undergo changes at different phases, depends upon your diet, your environment, even your thought process. So all of us start our journey on this planet as a single cell in our mother's womb as a zygote. The zygote contains half the chromosomes from the father and half the chromosomes from the mother, and we get 46 chromosome genotype, the zygote. And then the single cell becomes 2, 4, 16, and so on, and a 38 weeks fetus is formed and a child is delivered. So we have a journey from DNA to zygote to, and then fetus, and then human. So this journey is fantastic. I think I call it the greatest miracle in the universe of a human being born out of a single cell, because the human being is an animal with consciousness, with mind, and it whatever is being changed in this planet, good or bad, is because of that mind. So God has written life with 4 chemical molecules, that is adenine, guanine, cytosine, and thymine, we call ATGC, and this is 3.4 billion of them in each molecule of our nucleus. So basically, a baby is born, a boy or a girl, and the moment a baby is born, then the parents as well as the relatives start looking at the phenotype, whether the nose or the eyes or the hair or the skin color, what is the baby resembling at? Is the baby resembling the mother more or the father more? The height is like the father, the hair is like the mother, or the skin tone is like the family. All this comes into play. And then we have concepts, different symptoms, and then these symptoms we can today's age, we can correlate with the DNA. We call the DNA or the genotype. So we have a phenotype and a genotype combination. But today's world, the genotype, the DNA, is being challenged by different factors. There are external factors. We have sunlight, air pollution, water, the viruses, chemicals, infectious agents.
Sir,
and
sorry for the interruption. The slides are not changing.
Nothing, no slide is changing?
No, it is on the first slide only. Your name is showing.
Oh my God, you must have told me before. But
that's why I'm wondering. It is also not showing in the full screen.
And it is also not showing the full screen, Dr. Banerjee.
No, just now I have made it half screen, but it should change.
Dr. Banerjee, you have to manually change one by one. There is some, yes, now it is changing. Yes, perfect.
Changing now?
Yeah. Perfect.
But it is not showing the full screen.
No, when I go to full screen now, you can see?
Yes, it is the second slide.
Third slide?
No, you have to manually, yes, now it is changing.
Okay. So what I do is, I will keep it a little bit higher.
Yes.
Then I'll change like this. Because maybe there is some issue here in the change format.
Okay, no issues.
You can see the slide?
Yes, yes.
So I have told about my city. I have told about the transition. You have seen the human genome thing. Okay, you have, I am in the genotype slide, and this, okay. You can see the slide?
Yes, sir.
Yes, sir.
Okay. Right. So I was talking about various external factors, infectious agents, pollution, water, food. I'll come to food later because that's my topic. But I'm just giving you building up a story of what are the factors which can influence DNA 24/7 all the time. Now, there are external factors, and then there are intrinsic factors. There are intrinsic factors like congenital, you're born with certain problems, metabolic factors, degenerative diseases, neoplastic condition, immunological imbalances, hormonal imbalances. There are intrinsic physiological factors which can change your DNA. Then there are external factors as simple as temperature, humidity, pressure, stress, trauma, electricity. There are different anything and everything under the sky can have an effect on your DNA. People think everything hereditary is DNA, but I will say DNA is a dynamic molecule which can be affected by any smallest of the change which you undergo. Okay. So we consider damage to the genome as a fundamental disease that can be diagnosed and can be prevented. So any change on the DNA may damage the basic functionality of the DNA, which can be prevented, and also it has to be first diagnosed. So in aging, if you see, you have a lifespan. The human lifespan is about 100 years. So people live about 70, 80, 90, 100 years. That's called lifespan. Now, if we can monitor our genome health at various stages of your life, we can convert the lifespan into a health span. It is not important how long you're living in a lifespan, but it is very important how well you're living. And there, detecting changes in the DNA or stability of the molecule is very important. So DNA breaks life and causes diseases. This is fundamental, now proved by a lot of scientists around the world. I'll show you a few evidences during my talk. Now, all of you must appreciate that life is not happening to you, it is responding to you. It is not that, okay, this is happening, that is happening. It is responding. It's responding. Even we have a virus around the world, that's a response of 8 billion people living on Earth, which is not a healthy population. So life is also a competition of genes and minds. So there are some winners and there are some losers. It's always a competition. So Darwin had proposed the speciation, where the best and the toughest survive and the weakest die. It is all in the genes. So people thought that DNA can be used as a tool to detect, diagnose, and treat diseases. Precision medicine, which is the promise of precision medicine, is delivering the right treatment at the right time for the right person. This was declared by the American president, the then American President Barack Obama, in 2016. And then today we have the future is already here. We have a new field called the molecular medicine or personalized medicine of 21st century, where the medical world and the DNA world is slowly merging together, coming together. And why they're coming together, why this has happened? Because of the technological evolution. From last 20 years, from the early 2000 till now, we have moved from detecting single genes to panels to whole exomes and pathways. Today, we can see the whole genome. When I mean whole genome, the functionality of the whole genes which are functioning in the body, creating proteins, creating different processes, regulating different processes, can be profiled today. And this power of the technology has led to fields like cancer genomics, precision genomics, nutrigenomics, and infection genomics. So keeping that in mind, we created a platform called InDNA, which is looking solutions for life in DNA. And this is a company which I created in Bhubaneswar way back in 2012. My concept was a DNA clinic where we would look into solutions, and there will be a clinical approach, not only the problems, but finding solutions to the problem. And that's why we conceptualized a DNA clinic, which was new at that time. So what do we do here? We provide an integrated DNA-based platform for personalized medicine and molecular diagnostics. And we have been covered in different press forums as a very interesting platform where we can practice molecular medicine on a daily basis and work with clinical world. So we have moved from precision, from symptom-based or intuition-based medicine to pattern-based or evidence-based medicine. And very soon we will go into algorithm or data-based medicine, which is the DNA medicine. So we are actually undergoing an era, a sea change from generalized to personalized and to precision. So as everyone knows that people age differently, there are different, as I told you, genotype, phenotype. We can also call ageotype. People age differently because of their because they have good genes, they're good lifestyle, and there are different blue zones in this world where people are centenarians. They live beyond 100 years. So therefore, Okinawa is one of them. So there are different ageotypes. Why? Because this is purely because there are chromosomes which are integral part of your cell. In each chromosome, there are tails. In those tails, there are telomeres. And these telomeres get shortened every time the cell divides. So the way the telomeres shorten and the degree of shortening of the telomeres determine the age of a cell. And how the telomeres will go on will be the important fundamental programs of your cellular division and your cellular aging. Now, telomeres are a very important part. They can control the entire genome integrity, the function, though they are the tails, but they are very important crosstalk with the external cellular physiology and the genome. So telomeres are very, very important. That's why this field got this field is young relatively compared to the other field. The telomere field is only 25, 30 years old. But, every time a cell divides, so I told you that the cell, we divide from a single cell in our mother's womb to 10 trillion cells. So 10 trillion cells in your body has come from one cell. And that has been nicely monitored every time. Millions of cells divide. Right now it is dividing in your body. Telomeres are regulating it. If there are telomeric dysfunction, then there is segregation defects and genome instability in the body. So this is a fundamental discovery, and we have different diseases, most importantly cancer, because of telomere defects. And that's why this field had, Nature is one of the top journals in the world, and Nature had a cover page of Shiva, the god of immortality. And this cover page picture basically attracted me to this field about 22 years back. And I got interested in this field from this picture, which I saw in Bangalore NCBS library. And from then onwards, last 22 years, I've been researching on telomeres and genetics and how telomere regulates our body. So telomerase enzyme can stitch to telomeres back. Whenever telomeres get shortened, telomerase gets into functionality and stitches it back. Unfortunately, most of our somatic cells or grown-up cells do not have telomerase enzyme active. That's why we age and we die. Otherwise, we could have lived forever. So your genes and Janma is basically controlled by telomeres. That's how even Times Magazine has made it as a God gene. So this lady, whom I was fortunate to collaborate and work during my postdoctoral years, Carol Greider, has got Nobel Prize for her discovery in telomeres in 2009. And that's my name to fame in this field, and that's why I've been working in this field for the last 20 years. So yes, coming back to DNA damage, I was talking about DNA damage needs to be repaired. Thanks to our repair mechanism, that we are continuously repairing our DNA in spite of the damages we are doing. Even if you are having a Coke or a Pepsi, even if a Diet Coke you're having, your intestinal lining of your cells are getting damaged, and that is repaired. Some of them are repaired, or some of them die. So if the cells are not repaired, they need to die. That is called apoptosis. or necrosis. That is good, that is fine. Your body is programmed to do that. But if there is faulty repair, or if there is changes in the repair, then you have diseases like cancer. So if your body is under stress, your hormonal system, your immune system, your nervous system, if they are not in proper balance and there is an imbalance, we have a cell cycle deficiency, and that's why there is an arrest happening. Now the cells are not functioning properly, and the cells are under stress, and that's why the genome undergoes mutation changes. Now I'm coming back to this slide had animations, but since I'm not in that full slide mode, I can't show animation, but I can show you the DNA methylation, the change in the DNA. So the DNA molecule is a chemical, and the DNA molecule needs to be the DNA molecule the DNA molecule undergoes a chemical modification. What do you mean by chemical modification? The DNA undergoes an attachment of methyl group, methylation. So methyl group adds to the DNA and makes the DNA silent. It doesn't make a protein. So that is called the epigenetics, or the epigenetic modification, the environmental modification. There are two fundamental epigenetic modifications. One is DNA methylation and one is histone acetylation. So when methylation happens, the DNA becomes silent. But the same methylated DNA, when it is acetylated, an acetyl group happens, then the DNA becomes active, or the protein is made. So when the DNA is shut, it is methylated. When it is acetylated, it opens up and makes protein. So I'm not going into the complicated method of teaching today, but here you see that there are certain genes which are inherited from parents to the offspring. They're called intergenerational transmission of genes. But after they're inherited, they undergo environmental changes of methylation or acetylation during the lifetime. And these methylations, acetylations, and deacetylations can happen because of your exposure to different environmental conditions, including your diet. So, are nutrition professionals already practicing epigenetics? The answer is yes, but without the actual knowledge of epigenetics, without actually going into the chemical process of epigenetics. You are recommending a rainbow diet. What is a rainbow diet? All colored diet. Tomatoes, apples, turmeric, garlic, cinnamon, broccoli. All these are the colored foods. They all have ingredients which are vitamin boosters, which are micronutrient boosters. And now they are basically epigenetic modifiers. They can modify histones, they can modify DNA methylation. So that is what we will study now. So there is a healthy aging process. There is an inflammatory aging process called inflammaging. And this happens because some of your food can add methyl group to your DNA, and some of your food can add actually acetyl group to your DNA and make the DNA silent or make the DNA active. So the dietitian today's world with the technology can design customized nutrition therapies and lifestyle changes for healing, and also rather than managing symptom-based dietary management. So the anatomy of gene-diet interaction today is basically the DNA diet strategy. Let me see if I can show you this slide. Can you see this slide? Can you see the full slide, this one, everyone?
Yes, sir. Yes, sir.
This is very important.
It is not the full slide. It is not the full slide.
Can you see the full slide now? No?
No, sir. The slide is visible, but just not full screen.
Okay.
Full screen is not visible.
Okay, let me delete this. Yeah, now you can see, right?
No,
Okay, so
throughout the presentation, we could not see a single full screen. We always
no,
saw
that's
the
fine.
side.
I know. So now what I wanted to take off that, because this slide is the most important slide. Why? Because this is for understanding the anatomy of gene diet interaction. this is the crux of my talk. So when you are in the current diet of an individual, basically this diet the nutrition of this diet exposes the genome, and it creates epigenetic alteration impacting the expression of genes. Some of the genes get silenced and some of the genes get activated. Now, when I say activated, they can be activated for good cause, they can be activated for bad cause also. Now, this is epigenomic modification because of the diet. And that modification also is helped by some ethnic or ancestral or racial. Suppose you have a hereditary gene which modulates your cardiovascular metabolic effect, or you have a parents with diabetes, okay? So those the racial or ancestral history will add on to it. So genetic variants will be impacting, and the diet load together will form molecular phenotypes in your body. And because of these molecular phenotypes, people suffer of diseases, right? And that's why this is the anatomy of gene-diet interaction. How do we see it? Now, we do patient database collection. We do database of the patient. We take genome information database. We do mutational analysis. Then we integrate with the health, and then put our gene list, classify the gene list for high risk factors and low risk factors. So coming back here to real life scenario, suppose there is an obesity, and obesity is having with hypertension plus diabetes plus cardiovascular. These are very common. I'm not talking any hypothetical situation. The real world scenario, a obese person may be hypertension, may have diabetes, may have blood pressure issues, and is prone to cardiovascular. And now all this is also every time is activated by junk eating, lack of exercise, smoking, alcohol abuse, genetic and environmental factors. So this entire mixture is called the cocktail mixture of your mutation bubble we are in. We are getting changed. Our DNA is mutated by this entire bubble of unhealthy lifestyle issues which we are suffering from the current age. So coming back to another important term which you will see in genetic reports called single nucleotide polymorphism. Remember, I told that DNA is made up of ATGC, the 4 base pairs. Now, in this single, if the single base, there is a spelling mistake in your sentence of a chapter, a book, this single spelling mistake may have a drastic effect, as drastic as the whole vitamin pathway may be affected. The whole metabolic pathway may be distorted, okay? Or an absorption of a particular nutrient cannot happen. So these are called single nucleotide polymorphic changes, or called SNPs. For example, SNPs on a particular gene, MTHFR, can lead to different vitamin or folic acid deficiencies. So in your body, there are up to 70% of all the enzymes are SNP dependent, and their activities are governed by small changes in the SNPs. It can be homozygous changes, like 2 genes pair, your father's gene and your mother's gene SNP can be changed, or any one of them can be changed, or both the change is not there, they're healthy, a wild type. So you have homozygous or heterozygous changes, or no changes at all. Now, you can have SNP IDs for different genes. I'm not going into the details. They look like this. And there are thousands of such SNPs of genes which can be detected and correlated with deficiencies. So nutritionally relevant genes which are well researched today are associated with your folate metabolism, mood disorders, estrogen metabolisms, diabetes, cardiovascular problems, and etc. So as I told you, methylation, which is just addition of a methyl group, which is 1 carbon 3 hydrogen group attached to your DNA, can make several changes. Now, this role is used in loss of digestive integrity, mood disorders, neural tube defects, endocrine imbalances, colorectal breast cancers, lung cancers, cognitive function defects, and even inflammatory diseases, thyroid functions, fertility, fatigue, okay, poor health, and even immunity. So all these conditions are dependent on methylations or epigenetic modification of the SNPs. Now I'm just showing you snapshots of these pathways. They're complicated pathways, but it is not your job. It is our job to look into these pathways. But this is what goes on in a laboratory. I'm giving you a snapshot of what goes on in a laboratory. What kind of pathways do we study when we study a gene? And these genes are, each gene code for different enzymes, and those enzymes are associated with your dietary metabolism. And these enzymes also need cofactors like B12, B6, zinc, magnesium, and these cofactors are also used for your metabolic functions. And some of the SNPs or single nucleotide polymorphism can affect the use of such cofactors. Now, nutrients can bypass some of these SNPs. Suppose you are born with these SNPs. What do? We can't change the SNPs, but we can change them with nutrition, with different kinds of supplements, with different kinds of extra supplement. That is the work of a dietitian. And that's what targeted gene diet comes into play. And this is where you will see the functionality. For example, MTHFR. Hello? There are some people's mic is on. Can I request people's mic to be off? Okay, so MTHFR gene is important gene. I'm just taking one example, which is actually, if it is reduced, if there is an impact on SNPs of MTHFR, there is reduced conversion of folate to active form of the folate. And because of that active form, this C677T mutation, which is very common, there is a reduced conversion of homocysteine to methionine, and there is homocysteine accumulation in the bloodstream, which leads to cardiovascular problems, infertility, and many other diseases. So if there are medical histories, different signs and symptoms, we must correlate the biomarkers with those histories, particularly environmental toxin exposure, cigarette smoking, and the stresses like emotional and stress and psychological stresses, so that it gives us actual clues and correlate the biomarkers with. And there are different conditions of undermethylations. If there is an undermethylations, you can have cardiometabolic syndrome, you can have peripheral neuropathy, you can have different immunities and cancers. So low folate, low B12 backgrounds are again results of undermethylations. There are methylation issues where even urinary estrogen metabolite panels which are high can be correlated with the methylation problems in some of the SNPs. Drugs interfere with folates, and they absorb their absorption and availability of active form are important. Now, some of the drugs which are used on the clinical drugs like antacids and H2 blockers and proton pump inhibitors, anti-seizure drugs. Now, these drugs basically they may be prescribed for certain conditions, but they modify the genome, which may really lead to other diseases. That is important. So drug exposure history is extremely important for dietitian. So if the dietitian is asking drugs, means they are actually correlating that with the drug deficiency, with the dietary intake and dietary deficiency, because some of the drugs directly interfere with folate absorption and availability. So this metformin, colchicine, antacids, anti-seizure drugs, methotrexate for chemo in cancer, estrogen drugs, the sulfur drugs, they directly interfere with B12 absorption. So therefore, the diets are directly affecting your methylation status of the genome. I'm just giving you proofs. And there are some of the companies abroad like Nutrigenomics, they give this kind of reports, genetic clues for SNPs. And how do they add up in nutrition intervention? They try to correlate the genetic condition and give nutrition intervention to reverse or bypass these SNPs, and that bypassing mechanism can help integrating the dietary supplements when the sleep conditions actually give you a clue that these are the problems in your metabolic pathway. So those pathways can be bypassed by supplementing or changing the diet. So if you bypass the MTHFR gene, there is greater need for folate, active form of the folate, then folate-rich foods you can be given, particularly like liver spinach kit and other things. You are dietary experts, I need not explain that, but yes, they are needed for correlating with your condition. If you do not know the condition, then again it is a symptomatic treatment, not a directly nutrigenomic treatment. So targeted nutrition support is the way forward in this current era. And why diet may not be enough, inability to convert a vitamin form into active food form, you need supplementation, you need micronutrients, you need overall complete comprehensive approach, not only the change of diet, and also the support system like lifestyle changes, which are very important in metabolizing those. This is how a nutrition assessment is done. There's another protein, another kind of gene which I would like to tell you is the catechol methyltransferase. This catechol methyltransferase or COMT gene is another very important gene which actually detoxifies dopamine, epinephrine pathway. This is basically mood stabilizer. It changes the mood, it changes your behavior, and it is directly affected with your digestive system. So sleeplessness, excessive stimulation, alertness, wakefulness, restlessness, these are all associated with this gene. And if there's a mutation in this gene and that is detected, you can give more foods rich in vitamin B12 or multivitamins, and you can have detoxification, which Dr. Sri Kumar is expert in India and nationally renowned. So those things directly help these conditions. So this is some of the examples I would like to tell, which are important. There are more genes with nutritional relevance, which I'm not discussing today, but there are lists can go on. There are many lists, and also the gut biodiversity. There also some of the genes play a big role. So gut microbiome, change in the gut microbiome must sometimes directly affect the health. So there's a huge list which I can show you, which are related to the nutrigenomic relationship with genes. So last part of my talk, the last 5 minutes, 2 minutes, I can tell you that we are actually seeing only one part, that is the 3/4 of the problem is down. It is not beyond our perception. So we have to, as a clinician or psychologist or dietitian, we have to go beyond and look at the emotional impact. The mind plays a big role. And so why? Because psychological factors actually predispose a human being for poor coping group, and psychoneuroimmunology I showed you plays a direct role in your DNA. And there are conditions which are mild to severe, and that conditions needs to be mapped and that needs to be addressed. Because if you are under stress, actually the natural condition of adaptation goes off, and you are not able to adapt how much ever supplements and therapy is there, but adaptability changes. So we have to change the stress and trauma around us, that is the condition of the mind. So genome health assessment, comprehensive genome health assessment is an important tool today in the modern world to detect pathways, genes involved in lifestyle disorders, in different conditions of obesity, hypertension, diabetes, cardiovascular conditions, hormonal imbalances, accelerated biological aging, and many other non-communicable diseases. The list, as I told you, are long, and we can create different genome health index for people. We can create different nutritionally relevant, DNA relevant diet for them called the Nutri diets, which depends upon the genotypes of the people. And as I told you, the genes and the behaviors and life are all interconnected. They cannot be separated and treated isolatedly. They have to be a comprehensive view of it. We have to integrate the entire system. Without integration, we cannot come to proper diagnosis and managing the health. So Creation of topological maps for different platforms are important in human life. Why? Because whatever patient you are seeing, you're just seeing the tip of an iceberg that I showed you, because subclinical conditions or unseen diseases are apparent, are at large in the population. So diagnostic tests or few gene tests will only isolate few population. But if you actually go deeper, we will be finding more and more subclinical conditions. So from reactive health, we have to go into preventive health. And preventive health is where the whole crux lies. So what is the future of nutrigenomics? It will evolve from providing clues to underlying root causes to offer better predictive tools. Will ready us for the earliest nutrition intervention to treat diseases. That's what I call preventive management, and it will be very precise and targeted way. And the framework will evolve, and we will have personalized diet and lifestyle plans for optimal health and preventive strategies before diseases present. So this is the future. You are going to have first-hand knowledge, prior knowledge before the diseases comes and knocks your door. I would like to thank my collaborators from around the world. I have been fortunate enough to collaborate with few great giants in different areas of therapy, including Nobel laureates, where I seek inspiration, and my greatest inspiration is from none other than Swami Vivekananda. I take risks in life, and if I get success, I tell people, but if I lose, I guide people. That's not the way. So this is my team, and thank you very much.
Thank you very much, Dr. Banerjee. It was really, really very enriching for us.